A very good morning to all students. We are at the end of the Cell the Unit of Life chapter. The last topic, nucleus. Today we are going to discuss. So, and small microbodies also we'll discuss today. So, nucleus is a very important topic. In the cell, the unit of life, completely you see at least one question from or one or two questions from the from the nucleus. So this nucleus plays an important role in the cell because as it is the genetic material. So inside the nucleus you see the genetic material DNA. Okay, right. So who discovered the nucleus for the first time? So Robert Brown. So nucleus, no doubt, the nucleus was first discovered by Robert Brown. Okay, Robert Brown in the year 1831. Okay, so as I already said before, the largest cell organelle is nucleus. The first largest cell organelle is the nucleus, followed by second largest is chloroplast. The smallest is ribosomes. Okay, that's what we discussed in the previous classes. So largest dynamic cell organelle. Largest dynamic cell organelle which occupies 20% of cell mass. Occupies 20% of cell mass. Okay, right. Next, this controls and coordinates various types of activities. It is a spherical structure. It is spherical structure which controls and coordinates. Spherical structure which controls and coordinates the various life activities. Various life activities. Okay, right. Next, so this is, you can observe this nucleus only with the staining. The genetic material inside this is nothing but the chromatin. So chromatin can stain by basic dyes. Inside the nucleus contains chromatin. The nucleus contains chromatin material which is a long thread-like structure, long thread-like coiled structure which is a chromatin material. This can stain by basic dyes. Which can be observed by staining. which can be observed by staining, that is with basic dyes. Stain by basic dyes, stained by the basic dyes. So this was proposed by this staining was proposed by Fleming. The staining was proposed by Fleming. Okay. So what is that stain? Acetocaramine or fulgen. Basic dyes, acetocaramine or fulgen. Acetocaramine or by fulgen stain. So this is the stain where you can observe the genetic material inside the nucleus. Okay, right. Next, so this was generally absent in the mature RBC sieve tube elements, and that is in the vascular sieve tube elements of vascular plants. So nucleus is absent in the mature RBC sieve elements. Note down, nucleus is absent in RBC and sieve tube cells or sieve tube elements. Sieve tube elements. Okay, right. So there are the nucleus is a double membrane. It is the covered by a double membrane. You can note down. It is nucleus. It is covered by double membrane structure, which is of 5 to 10 nanometers thick. Which is of 5 to 10 nanometers thick. Okay, right. Next, it is separated by perinuclear space. So double membrane is separated by the nuclear membrane. The nuclear membrane is separated by space, that is called as perinuclear space. That is perinuclear space. And this space, if you see, this is of 10 to 50 nanometers gap. 10 to 50 nanometers space. The space is of 10 to 50 nanometers space. Okay. So this is with related to this. And you already know that the outer membrane of the nucleus is attached to the endomembrane structure called endoplasmic reticulum. Okay. So we already know that the endomembrane reticulum is attached to the outer membrane of the nucleus. The outer membrane is attached to endoplasmic reticulum. Outer membrane is attached to endoplasmic reticulum. Next, the pores. Outer membrane has pores that is called as nucleopores, nuclear pores. Primary attached to the reticulum and consists of pores called nucleopores. And called as, these pores are called as nuclear pores, okay, which is the pore size is 10 to 100 nanometers. Right. Next, this the diameter of this nucleus is envelope is 10 to 100 nanometers, diameter of the nucleus. Diameter is 10 to 100. Next, nucleoplasm. The fluid material, what you call it as a nucleoplasm. In the inside the nucleus, the granular fluid material inside the nucleus the granular fluid material called nucleoplasm. Called nucleoplasm is present. Nucleoplasm is present. Okay, right. So this is consists of this nucleoplasm consists of genetic material DNA. Consists of genetic material DNA. Okay, right. And also that is nothing but in the form of chromatin. DNA means chromatin and nucleolus. Chromatin and nucleolus. Okay, so nucleolus is nothing but it is also a darkly stained spherical structure. What is nucleolus? Spherical structure, darkly stained, which can able to synthesize. So this can able to synthesize ribosomes. That is rRNA, ribosomal RNA. Nucleoplasm that contains the ribosomes. It is a site of rRNA synthesis which synthesize rRNA, ribosomal RNA, rRNA synthesis. Next, they generally, what is the function of this nucleus? It coordinates functions of different cell organelles. So it is called cell brain or dynamic center of the cell. The nucleus is called cell brain or dynamic center of cell. Dynamic center. It is the dynamic center or cell brain. Okay. So the function is it coordinates the function of different cell organelles. Nucleus coordinates the function of different cell organelles. Different cell organelles. Okay, right. So the transfer of genetic information, generally the transfer of genetic information. Transfer of genetic information. Okay, so this is what generally we see in the nucleus, and we should also discuss about the chromosomes. The chromosomes are, so the chromosomes also part of the nucleus. Part of the nucleus. So chromosomes these are observed. Chromo means color, zome means body. You already know. Chromo means color, zome means body. Chromosomes. So these are observed first by Hofmeister scientist, and generally this is consists of the chromatin is formed. The chromosomes are formed from the chromatin. The chromatin condenses to form chromosomes. Generally, the chromosomes are formed from the chromatin material, which is a thread-like structure. So generally, the 2 meters long thread distributes into 46 chromosomes. The 2 meters long chromatin distributes into 46 chromosomes. That means, how does the chromosomes will be formed? From the chromatin material condensation. The condensation only will form the chromatin material, which is generally 2 meters in long. The length of that in humans, if you take the length, it is approximately 2.2 meters generally. So 23 pairs of chromosomes we are calling, no, 46 chromosomes, nothing but 23 pairs of chromosomes. So these are formed by the condensation of the chromatin material, and then they distribute into 46 chromosomes. Okay, yes, these chromosomes can generally absorbed by staining, what we have already discussed, right? So if you see this, it is generally the shape of this will be circular in the prokaryotes and in the linear in the case of eukaryotes. And the chromosome consists of thread like, that is it is having the different structure. So based on its metaphase, we can easily study the structure of the chromosomes. In the metaphase of the cell division, we can have the structure of the chromosome, that is the chromatids. These are called as chromatids, centromere. Okay, and each chromosome consists of two chromatids. Each chromosome consists of how many chromatids? Two chromatids, and totally 4 arms. Arms are 4. 4 arms. Okay, and this chromosome has a centromere, that is primary construction. The centromere is nothing but the primary construction of the chromatids. The primary construction of the chromatids, we call it as a chromosome. So chromatids, these chromatids are nothing but longitudinal half of chromosome. It is a longitudinal half of the chromosome, and each centromere has kinetochores, 2 kinetochores. Each centromere has how many kinetochores? 2 kinetochores. Each chromosome has how many chromatids? 2 chromatids. Okay, right. And this, let us see about the detailed information about this structure and also classification of the chromosomes. Okay, right. So the chromatid, note down, what is this chromatid? So you can see where is the DNA then? This chromatid consists of the DNA. 2 strands of the DNA which are close to each other, that is double stranded. That means, what is chromatin and DNA difference? This chromatin is associated with histone proteins. So here if you take DNA and histone proteins together, we call this as the chromatid. The genetic material DNA and histone protein associated together is called as a chromatid, whereas the DNA associated with the histone proteins, we call it as chromatid. So this chromatid is a longitudinal half of chromosome. Chromatid is the longitudinal half of chromosome. It is the longitudinal half of the chromosome. It has 1 DNA molecule. It has 1 DNA molecule. The 2 chromatids are attached to centromere. The 2 chromatids are attached to centromere. So they are attached or constricted to the centromere. So having 4 arms. Okay, 2 chromatids attached to centromere, which appears as 4 arms. Next, the chromatin is the chromosomal material appear as a filamental structure. So chromatin is the single linear DNA and histone proteins. Single linear DNA DNA and histone proteins. Together we call it as a chromatin, okay, which is the thread-like structure. The centromere is formed. The chromatids are connected at the centromere, which is by primary construction. Okay, centromere. So the centromere is formed by the primary construction. The centromere is formed by the primary construction and divides the chromosome into arms. The centromere divides the chromosome into arms. This divides the chromosome into the arms, and it is a non-stainable part. Very important. Centromere is non-stainable. The centromere is non-stainable. It cannot be stained, so it cannot be observed. It cannot be stained or cannot be observed. And inside the centromere, we see the disc-shaped structures called as the disc-shaped structures, proteinaceous structures surrounds the centromere, that is called kinetochores. What are kinetochores? The proteinaceous disc-shaped structures surrounds the centromere. Surrounds the centromere, we call it as a kinetochores. Okay. So spindle fibers are attached to the kinetochores during the cell division. Generally, spindle fibers are attached to the kinetochores. Spindle fibers are attached to the kinetochores during cell division. Are attached to the kinetochores during the cell division. That is with the kinetochores. Okay. Next. The secondary construction, again it can have also a secondary construction just like in primary construction at any point besides primary construction. Continue. Secondary construction besides primary construction, chromosome can have secondary construction. Have secondary construction. Okay, right. So this is also non-stainable and lack kinetochores. This lacks kinetochores and non-stainable. So it cannot be stained. non-stainable, and lack the kinetochores. So this chromosome, what it is having, the secondary construction beside the behind the secondary constructions, if you see, it appears like a knob-like structure called satellite or sat chromosomes, non-stainable. They have a secondary construction and appears as knob, called sat chromosome, that is satellite chromosome. SAT stands for satellite chromosome. Such kind of chromosomes are called as satellite chromosomes, which is non-coding. It cannot code, it does not have any genetic material, it is a non-coding part of the chromosome. Okay, so round knob-like structure beyond secondary construction is a round knob-like structure beyond secondary construction. Beyond secondary construction, we see a round knob-like structure. Okay, right. So this is nothing but the non-coding and forms heterochromatin. This sat chromosome is non-coding part. It cannot code for any information. Non-coding. You'll understand this non-coding and codings, heterochromatin, all these in the molecular basis of inheritance chapter. Okay, is non-coding and heterochromatin. So it cannot undergo any transcription. Euchromatin and heterochromatin means heterochromatin, it cannot undergo any transcription or form any proteins or enzymes. And next, the ends of the chromosomes have polarity. Chromosome has polarity. The ends of the chromosomes has polarity called telomeres. These are called as telomeres. So what does they use? They prevent the fusion between the two chromatids, which prevents the fusion of two chromatids. So if the chromatids are fused, they form into a single chromosome. So that fusion it will prevent two chromosomes or chromosomes. Prevents the fusion of two chromosomes. Next. So coming for the classification of these chromosomes, classification we have the based on the position of the centromere. Classification based on the position of the centromere. Okay, yeah, based on the, just a minute. Right. So classification which is based on the position of centromere. Based on the position of the centromere, the chromosomes are classified. The chromosomes are classified as different types. They are metacentric, submetacentric, metacentric, submetacentric, acrocentric, and telocentric chromosomes. Metacentric, submeta, acro, and telo. Metacentric has it is nothing but equal half set. It forms equal halves. The chromosome, the chromatids are of equal halves. Equal halves. So it is V shape. It exhibits which shape? V shape. Whereas if you take submetacentric, centromere away from the center. So here centromere is at the center. Are equal halves. Centromere is at the center. So only it exhibits the V shape. Whereas in the submetacentric, centromere away from the center. Centromere away from the center. So it exhibits a L shape. Which shape? L shape. Away from the center, L shape. So centromere close to the end, J shape. Centromere close to the end, J shape. Centromere close to the end, J shape. Whereas at one end, centromere at one end, centromere at one end, that exhibits I shape. So V shape followed by L shape and J shape, I shape. So this is metacentric, submetacentric, acro, and telocentric. Okay, so these are the classifications of chromosomes based on the position of the centromere. Is it clear? Right. So this is about the nucleus and chromosomes. So hope you understood this nucleus and chromosomes. So there are in detailed explanation about of NCERT. Also some points are there, so you can revise those things. And so we have a small topic called as microbodies. So that microbodies, you just keep the heading as microbodies. Okay, that also will complete. With that we'll complete this particular chapter. Microbodies. So what are these microbodies? So membrane, many membrane bound minute vesicles. Microbodies. Microbodies are many membrane bound minute vesicles. So called as these minute vesicles which contains various enzymes which contains various enzymes in both plants and animals. In both plants and animals. We call it as microbodies. Okay, so it consists of, what are the examples of microbodies? Spirosomes. Examples of these are spirosomes, glyoxysomes, glyoxysomes, next lomasomes, peroxisomes. Lomasomes, L-O-M-A, lomasomes and peroxisomes. So these are about these microbodies. Okay, right. So this is lipid synthesis. Spirosomes are nothing but lipid synthesis. Lipid synthesis. These are fats are hydrolyzed. The fats are hydrolyzed generally to the lipids. Fats are hydrolyzed to acetyl-CoA lipids. So lipids to carbohydrates. Glyoxysomes. Lipids to carbohydrates. Next, lomasomes. It is present between the cell wall and cell membrane, function as a wall formation. It is function as wall formation. They function as a wall formation present between the cell wall and cell membrane. Peroxisomes. It consists of catalase and peroxidase. Catalase and peroxidase. Okay, catalase and peroxidase. So these are the functions of the microbodies. Okay, right. So with this we completed this particular chapter. The next class we will start a new chapter. Okay, thank you.